Hi everyone, this is our second lecture on variations in Mendel. Last time we covered incomplete dominance, codominance, and lethal traits. This time we're going to be talking about some other examples where Mendel's laws originally didn't seem to hold up. So let's look at some questions. The first question is this one. The phenomena where not all individuals within the genotype show the phenotype to the same degree is referred to as what? Second question, if an individual allele has more than one effect on the phenotype, it is said to be what? And number three, the interaction of non-allelic genes in forming the phenotype is called what? The first concept here is one we were introduced to already in the PDX-1 assignment. It's called incomplete penetrance. And this is seen when individuals of the same genotype don't all express that phenotype similarly. This is shown here on the left-hand side with wheat. The wheat kernels are brown or white. The brown is the genotype, but for some reason, some wheat kernels are not expressing that, and so they appear white. This is called incomplete penetrance, where individuals of the same genotype are not expressing the phenotype. The reason for this could be varied. For example, impacts from the environment, impacts from the genetic background of the organism, so the other genes, the other alleles that are essentially not being accounted for in this cross. So there's a variety of reasons why that can occur, but it's called incomplete penetrance. An extension of that is called expressivity. Here it is in the middle, where all the wheat kernels are the same level of brown color. This is called constant expressivity. However, if the wheat kernels are at different shades of color, this is called variable expressivity. Again, it's a sort of an extension of this idea of incomplete penetrance. Other genes, what we call the genetic background of the organism, can be influencing how that brown color is being expressed. Impacts from the environment could be impacting how that brown color is expressed. We don't know. On the right-hand side here is a combination, incomplete penetrance with variable expressivity. You will see this not just with wheat kernels, obviously, but for example in diabetes. Individuals who have the same genotype may not all develop diabetes equally. Some may only have glucose intolerance. And the reason for that is, well, because the genetic background of that individual, the other genes that are involved, may be impacting how they metabolize food, how they respond to insulin, for example. And then there's the environmental contributions, diet and exercise. So, what causes incomplete penetrance? Like I said, there's genetic background. You can have effects from other genes that would be included in the genetic background. For example, some other genes that are not part of the cross might be suppressing the expression of the one that you are looking at. An effect called position effects also occur sometimes. Position effects are where the location on the chromosome affects expression. For example, here are two flies that have the same genotype for the eye color red, and yet you can see that their eye colors are not the same. The color on the fly on the top has the normal red eye color, but this fly is heterozygous, red and white. Red is dominant to white, and so the fly shows up red eye color. The fly on the bottom, however, has had something called a position effect. The red gene has been moved onto a different chromosome where it is now suppressed, and the white gene, the white allele, is now showing to some extent. So the red allele is still showing, but severely impacted, and the white allele is now coming through. So same genotype, but the position of the red allele has been moved. Here's another example of non-Mendelian traits. It's called pleiotropy. Pleiotropy is when a single gene mutation causes a range of phenotypes. This example here is called porphyria, but we've also seen sickle cell anemia as another example, and Marfan syndrome, a problem of connective tissue, as an example. Porphyria is a problem with blood development. Porphyria doesn't allow heme production correctly, and so it's a blood disease. Blood, as you can imagine, impacts lots of different organs throughout your body, from the brain to all the internal organs to everything you see here. So one gene mutation can cause all of these symptoms: abdominal pain, constipation, limb weakness, and so on. Missing enzyme porphyrin accumulates in the blood, in the red blood cells, doesn't allow oxygen transport to happen very effectively, and so it leads to lots of systemic problems. It's hard to pin down this into like a nice, neat 3-to-1 ratio if you had two people who are heterozygous for porphyria, because of the complex nature of all of the symptoms. So that's pleiotropy. One gene mutation, but lots of symptoms, not a single trait. Some environmental impacts can influence how genes are expressed. And this is something Mendel also did not discover. Here's an example of a Siamese cat, where they possess an enzyme that makes a pigment. This black pigment is produced in the face, the ears, the extremities, and that black pigment is produced by an enzyme that is temperature sensitive. It produces this enzyme produces the black pigment only in cooler parts of the cat's body, the extremities, the nose area, the ears. In the core part of the body, where the white color is, the enzyme is slightly unfolded by the increased temperature and doesn't work properly. And so it doesn't produce that black pigment. So this coloring is due to a single gene mutation, and yet it doesn't express uniformly. You'll also see this with, for example, the Himalayan rabbit. Nutritional effects are another example of environmental effects. Lactose intolerance, like we talked about. Again, you may be lactose intolerant, but if you never drank milk, you wouldn't know it. So it depends on your interaction with the environment for how the trait is expressed. Exposure to chemicals or radiation can lead to things like freckling, but freckling is not something you can easily quantify. For example, two twins who are prone to freckling will have different patterns of freckling. So it is environmentally influenced. Phenylketonuria is another example. Phenylketonuria is a disease where individuals cannot break down the amino acid phenylalanine. Some foods contain excess phenylalanine, and that can build up in these patients' bodies to lead to problems. So you'll notice that a lot of food products, sometimes even soda cans, will have warnings for phenylketonuria to be aware that this product does contain phenylalanine. But again, if you never drank that product, you wouldn't know that you were affected. What about sex and gene expression? There are some traits called sex-limited traits that only are only seen in certain sexes, for example, females or in males. Examples include production of milk in mammals. Both males and females harbor the genes for milk production, and yet only females express them. Again, due to the genetic background of females, meaning the other genes that influence the production of the milk. Horns on sheep are an example. Both sexes express the horns, but the size of the horns is much different, influenced by the male genes. Another type of sex trait, effect of sex on gene expression, is sex influence traits. For example, pattern baldness. Let's look at an example. Pattern baldness can be described this way, with a gene symbol B for baldness. Two carriers for this allele, this bald allele, shown here as the red allele, would have different expression of that allele in the sex. So females who are heterozygous have hair, but males who are heterozygous are bald. Again, it's influenced by other genes in the male or female genome. The gametes formed from these individuals would have either the hair allele or the bald allele. And then in a Punnett square shown down below, these are the possibilities that are represented. Individuals who have two of the hair alleles, the B plus alleles, would have hair. Heterozygotes, though, would differ on sex, either bald in males or hair in females. Inheriting two bald alleles, though, would produce pattern baldness in both the male and the female. Another example with two traits is called modified dihybrid ratios. A lot of times phenotypes are often affected by more than one gene. We saw this in the previous examples when I talked about genetic background and things like suppression, how certain genes that are not part of the cross you are conducting are influencing that cross. So this example here is where we're looking at a specific gene that influences the expression of that second gene. And this concept is called epistasis, when the effect of one gene masks or modifies the effect of another. Here's an example in coat color with mice. And this is an example of recessive epistasis. This is a wild type coat color, this kind of brownish color with flecks of black or gray. It's called a goody, and the allele that produces this coloring is called C. So capital C produces pigment, and capital C is dominant to little c, which does not produce pigment. So a mouse that has coloring like this would have the big C allele. There's another gene called gene A, which produces this flecked patterning seen here. And this is the agouti patterning. It gives you that kind of wild type look to a mouse. The big A allele produces this pattern. The little a allele does not produce the agouti pattern, and the mice look black. You can think of this as a biochemical pathway. Let's imagine that these mice produce a precursor molecule which is colorless. The gene C, for making the pigment, takes that colorless pigment, a colorless molecule, and changes it into a black pigment. The dominant C allele would make black pigment, the recessive C allele would not. Gene A then gets involved. It takes the black pigment and then modifies it to make the agouti patterning, and hence the epistasis. Gene A is modifying gene C. The product of the enzyme gene A is modifying the product of gene C. Here are some example genotypes. Mice that have dominant C and dominant A will produce the agouti pattern. They will be agouti. Mice that produce the dominant C but recessive A's will have black mice because the product ends with this black pigment. But mice that have recessive C, little c, do not make pigment at all. They are colorless and essentially albino. And that is regardless of whether gene A is dominant or recessive. For example, if gene C is recessive, there is no production of black pigment. If there's no production of black pigment, there's no chance for gene A to modify that black pigment to make it agouti. Hence, the albino mice could be little c with a dominant A or a recessive A. In either case, the mice will be albino. Okay, that's recessive epistasis, and I will see you in class.